Good morning, everybody. So I'm going to talk to you this morning about the National Nutrition Phenotype database. I'm going to give you an overview of this database, and I'm going to just pick out some examples of potential uses that we can put this database to use, and I'm going to have a focus really on personalised nutrition. So as we've sort of learned from Eileen's talk, if we are to move into the zone of personalised nutrition, we really need to start thinking about gene and environmental interactions. And the environmental interactions can include, but are not limited to, things such as diet, exercise, smoking, medication use. And Eileen has just highlighted to you some of the gene diet interactions with a specific emphasis on single genes. But if we really are to move into the field of personalised nutrition, we need to start looking at the integral sum of genetic and environmental factors. And this is where we believe that a nutritional phenotype database will help us, and will help us to move this field forward. So in developing a phenotype database, we envisage that it will have many uses, and I'm just going to highlight some of the uses that I'm going to talk about today, but the phenotype database is in no way limited to these uses. So in the first area, we do think that it's going to really help us in this field of development of personalised nutrition, and I'll talk a little bit about that today. We also think that it's going to have and play a big part in the development of biomarkers, new biomarkers for different areas, and I'm going to talk specifically about one area today. And it'll also help us in biomarker validation. So to set about organising a phenotype database, Mike Gibney formed a consortium called JINGO, which is the Giant Irish Nutrigenomics Organisation. And Eileen has already mentioned that this is a consortium of 4 universities on the island of Ireland. So it's UCD and Trinity College here in Dublin, it's University College in Cork, and University of Ulster. And this consortium then is building the National Phenotype Database, and it is funded through the Department of Agriculture and the Health Research Board. So traditional databases in nutrition research would collect dietary data, a little bit of biochemical data, a little bit of physical activity data, and some anthropometry data. What's different about the National Nutrition Phenotype Database is that it's also collecting not just these 4 types of data, but also a vast extra amount of data, such as a lot of nutrigenomic data, transcriptomic, proteomic, and metabolomic. The fitness data, the physical activity data in one of the cohorts is very detailed fitness testing and substrate utilization recording. The body composition is very detailed body composition. We also have genetic data, and we also have metabolic challenges in the form of oral glucose tolerance tests and oral glucose tolerance tests and oral lipid tolerance tests in one of the cohorts. So we have a vast range of data that is available to us in this National Nutrition Phenotype Database, and I'm just going to talk to you today a little bit about how we're going to use some of this data. So if we look along these axes, we're going to cover very well the dietary axis, we're going to cover very well the phenotype axis, and we're going to cover very well the genotype axis. So we're going to be able to look at the interaction between these 3 axes and really probe the interaction between them, and it's develop further the field of personalised nutrition. So the 3 cohorts that are forming part of the National Nutrition Phenotype Database are the NANS cohort, which is the National Adult Nutrition Survey, the TUDA cohort, which is the Trinity Ulster Department of Agriculture project, and the Metabolic Challenge cohort, so-called the MESH cohort. I'm going to talk primarily today about the MESH cohort, but I'll briefly mention the NANS and the TUDA cohort. So the NANS is, as I said, it's the National Adult Nutrition Survey. It's a survey of 1,500 adults in Ireland, and it contains detailed dietary data, nutrigenomic data in the form of metabolomics and proteomics, and some transcriptomic data. It also has genetic data on these 1,500 adults and physical activity data. The TUDA cohort then is a cohort of 6,000 elderly subjects, and it's split into 3 different sub-cohorts. 2,000 of these have impaired cognitive function, 2,000 impaired skeletal function, and 2,000 impaired cardiovascular function. And the final cohort is the Metabolic Challenge cohort and the MESH cohort, and that is carried out here at UCD. So following screening, we have 214 subjects aged between 18 and 60 years. who presented themselves 3 times to the research centre here in UCD. On 2 of those occasions, they underwent a metabolic challenge. On the 3rd occasion, they visited the Institute of Sport and Health, where they underwent a series of physical testing. So the subjects then were randomised to receive either 2 oral glucose tolerance tests, 2 oral lipid tolerance tests, or 1 oral lipid tolerance test and 1 oral glucose tolerance test. And during these tolerance tests, we collected blood, urine, and in some cases adipose tissue samples. And then in the physical testing, when it performed in the Institute of Sport and Health, the subjects presented themselves and we measured the resting energy expenditure. We then followed this by detailed body composition analysis. We formed a DEXA scan and a bod pod. The volunteers were then given a light breakfast, and we performed some strength and fitness testing on the subjects. So all of this data then is being collected together with the biofluid data and our nutrigenomic data. So for each subject within this cohort, we have really detailed information about each subject. But rather than zoom in on each individual level, we believe that we can make more progress at this area by considering clusters, and I'll come back to that in a few minutes. But for each subject, essentially what we have is we have genomic data, transcriptomic data, proteomic data, metabolomic data. On top of that, and what's unique about our database is that we'll also have fitness function tests, we'll have detailed body composition, we have the response to metabolic challenges, and we'll have dietary data intake. So we'll be able to examine the functional tests and the relationship between these different levels in a systems biology approach, the differences are the relationship between genomic and function, transcriptomic levels, proteomic levels, and metabolomic levels. So, as I said just earlier, rather than zooming in at an individual level and looking at an individual, we have a very large interest in looking at clusters of individuals. So grouping people into clusters that are based on similar biochemical profiles or similar dietary profiles. So we aim to use this database to enhance this concept and to look at this concept of grouping people into different types of clusters. And I'll come back and I'll give you an example of where we use that later. The second area that we were going to develop and use this database for is in the area of biomarker development. So biomarker development can be divided into 2 areas, biomarker discovery and biomarker validation. So the National Nutrition Phenotype database will play a key role in biomarker discovery, and one of the areas we're particularly interested in is in biomarkers of dietary intake. And we will have really a great opportunity within the National Nutrition Phenotype database to make advancement in the area of biomarker discovery for dietary intake. And I'll give you an example of that and how we're going to approach that in a minute. We'll also be able to use the database for biomarker validation. And biomarker validation can be done either in intervention studies or in cohort studies. And again, if biomarkers are discovered in a different cohort or in a different intervention, we can bring them into the National Nutrition Phenotype database and validate them within this database. So we envisage that the database would play a key role in both the discovery phase of biomarker development and in the validation phase of biomarker development. So just to give you a flavour of how we're going to approach development of biomarkers of dietary intake, I'm going to give you an example of a study that we've recently published investigating biomarkers of dietary intake. So this was a study we performed in collaboration with University College Cork, where we recruited 160 subjects, half in Cork, half in Dublin, half males, half females, and we collected 3-day food diaries. This was part of an intervention study, and I'll talk about the intervention study later on. So we performed dietary pattern analysis on the dietary data. So we took our food diaries, we entered them into our dietary software, WISP in this case. We identified 33 food groups, and we performed k-means cluster analysis on these food groups, and we identified 3 dietary patterns in our population. And we called these cluster 1, 2, and 3. So cluster 1 was characterised by a high intake of vegetables, a high intake of whole milk, and a high intake of wholemeal bread. Cluster 2 was characterised by high intake of yogurts, high intake of fruit, and high intake of eggs. And cluster 3 was our traditionally unhealthy Irish diet, which was a large intake of alcohol and a large intake of red meat and red meat products. So we can see that again in this table here. So this is cluster 1. We can see that it had a high intake of vegetables, a high intake of wholemeal bread and whole milk. Cluster 2 had a high intake of poultry, had a high intake of eggs, and a high intake of yogurts. And then cluster 3 had a high alcohol intake, a high intake of butter and spreads, a high intake of white bread, and a high intake of red meat and red meat products. So our question then was, were these dietary patterns that we found in our dietary data reflective in our biological samples? So we took a metabolomic approach, and we asked the question, were they reflective in our metabolomic profiles? And the answer was yes, they were reflective in our urine metabolomic profiles, and this is just to give you an example of that. So this is the partially square discriminant analysis of the subjects that were classified into cluster 1 and cluster 3, and we can see separation of cluster 1 up here and cluster 3 down here. So the interesting question for us then was, what is different in the urine of these subjects that consume a dietary pattern in cluster 1 and subjects that consume a dietary pattern typical of cluster 3. So we were able to find these metabolites that were different, and 2 of these metabolites were particularly interesting to us. One is O-acetylcarnitine, and the other was phenylacetylglutamine. So if we look at O-acetylcarnitine, it was higher in cluster 3, and phenylacetylglutamine was higher in cluster 1. And if we recall back, cluster 3 was the cluster that had a high intake of red meat, and cluster 1 was the high intake of vegetables. So if we look at our cluster, zoom in on our phenylacetylglutamine, and we look at vegetable intake across the total population, and we look at percentiles of vegetable intake, we can see a relationship between the vegetable intake and the concentration of phenylacetylglutamine in the urine, and it increases as vegetable intake increases. So if we look at the red meat example, again, if we look across our total cohort and we group our subjects into percentiles of red meat intake, as red meat intake increases, so does the concentration of O-acetylcarnitine in the urine. So what we have here are 2 potential biomarkers of dietary intake, one for red meat intake and one for vegetable intake. And if we do a ROC analysis on the O-acetylcarnitine, for example, we see that it has quite a high sensitivity and a specificity for red meat intake. So now we have these 2 potential biomarkers, novel biomarkers. Now we have the potential of validating them within the National Nutrition Phenotype database, and that would be validation within the NANS cohort. So we have our database, we have our detailed dietary data from them, so we'll be able to get our red meat intake and our vegetable intake. We also have biofluid samples from them, and we'll be able to now go and take our 2 biomarkers and quantify our 2 biomarkers in these urine samples and validate these 2 biomarkers within the NANS cohort. So this is just giving you a flavour of how we will approach some novel discovery of novel biomarkers of dietary intake. So we'll further develop this within the National Nutrition Phenotype database, and we'll also further develop this concept of a nutritype. So this is the concept that a profile or a combination of biochemical markers can reflect a dietary intake, and that people can be grouped into different nutritypes based on their biofluid profiles. So these 2 concepts we'll be able to further develop. So now the second example I want to give you is how we can use a phenotyping approach in a move towards really going towards personalised nutrition. And this is, again, going back to this idea, rather than zooming in on one individual, what we're going to do is we're going to cluster individuals into groups that are very similar to each other based on their biochemical profile. And I'm going to give you an example of how we've done this already in one intervention study. So this was the intervention study of the part of the study that I talked to you about earlier, and it was a vitamin D study. So our subjects underwent a 4-week intervention trial, either with vitamin D or a corresponding placebo, and we collected biofluids in the form of urine, blood and fecal samples prior to the intervention and after the intervention. So we were interested from the beginning of phenotyping our subjects based on biochemical measurements that we performed in the plasma samples. So we took 14 biochemical parameters. We performed a cluster analysis in the form of k-means cluster analysis, and we had identified 5 distinct phenotypes in our cohort. And we simply called these cluster 1, cluster 2, cluster 3, cluster 4 and cluster 5. And each one of these clusters had a distinct biochemical profile, and I'll show you that in this slide. So cluster 1 had characteristically high levels of CRP. Cluster 2 was a typical insulin-resistant phenotype. It had high levels of glucose, high insulin, high HOMA score, high C-peptide, and it also had a high leptin level. Cluster 3 had the highest 25-hydroxy-D levels. Cluster 4 had high TAG levels, and cluster 5 had low 25-hydroxy-D and high levels of the adipokines resistant and adiponectin. So each one of these clusters had a distinct biochemical profile. And the question we asked was, were any of these phenotypes or clusters responsive to the dietary intervention that we performed? So if we take one step back and we look at the dietary intervention as a whole, and we ask, was there any effect on the of the dietary intervention on the dietary intervention on all of the subjects? So here we're comparing the subjects that were on the placebo versus those in vitamin D with respect to 15 biochemical parameters, and the answer was none of our parameters in the whole population changed. However, if we went and then looked within each cluster and looked at those that were on placebo versus those that were on vitamin D3, we found that cluster 5 was a dietary responsive cluster. And if we look at cluster 5 then, and we compare those that were on the placebo group versus those that were on vitamin D3, we found that our insulin levels improved following in consumption of vitamin D3 for 4 weeks. HOMA score improved and the CRP levels decreased significantly at the end of the 4 weeks. So what we had here was a dietary responsive phenotype, or a vitamin D responsive phenotype. However, if you remember back to cluster 5 did have the lowest level of 25-hydroxy-D and the highest level of adipokines. So we had to rule out the possibility that we weren't just seeing a responsiveness because we had low levels of 25-hydroxy-D to start with. So we went back and we subgrouped our population according to the 25-hydroxy-D levels. So we grouped them into 3 groups: those with less than 50 nanomoles per litre, those between 50 and 75 nanomoles per litre, and those with greater than 75 nanomoles per litre. And then we looked within each one of those groups at the placebo group versus the vitamin D3 group, and we found, even in this group here, no significant changes in our biochemical parameters. So really our vitamin D responsive group was a group that had low levels of vitamin D and high levels of the adipokines. It wasn't simply due to the fact that they had low levels of 25-hydroxy-D. So it really needed a phenotyping approach combining all of the biomarkers rather than just one simple biomarker. We also had metabolomic data for these subjects, so we were able to zoom in on cluster 5 and look to see what happened on other metabolite levels pre and post the intervention. So this is just showing you these are the samples pre the intervention and these are the samples post the intervention. So we were able to look to see what parameters have changed. So we have a change in glucose, a change in lactate, a change in lipoproteins, and an increase in one of the amino acids, glutamine. So we will develop this concept further within the National Nutrition Phenotype Database, and this concept of the use of clusters or metabotypes. And we will use the database to really drive this concept further and to see how we can use it within the context of personalised nutrition, and it will be explored further. So just to finish off, there is a large international interest in phenotype databases, and there is an initiative led by Ben Van Omen, who is here in the audience, for national nutritional phenotype databases. And what Ben has been championing over the last few years is this idea that of a nutritional phenotype database, and it was launched last year, where you have an online open source database where you can store all of your nutritional, or all of your metadata and your nutrigenomics data. So you're really storing all of the data of the full pipeline of a nutrition study. And the concept within this is that you'll be able to share data between different partners, and eventually the idea is that this database will become a depository where nutrition studies could be available to the public, and where we'll be able to further understand and look into gene-environmental interactions. So just to sum up then, what we have here is a very detailed database. It contains a wealth of information. So it contains our dietary data, very detailed biochemical data, and very detailed physical activity data, detailed body composition data, genetic data, and on top of this, it's not just limited to measurements and homeostasis. We also have dynamic measurements in the form of responses to different metabolic challenges. So in summary then, we're going to be able to really examine the interaction along these three axes, so along the dietary axis, genetic axis, and the phenotype database. And we will hopefully make advancement in biomarker discovery, in the use of clustering or metabotyping of individuals in personalised nutrition. So that just leaves me then to acknowledge all the people involved. So the JINGO consortium is, as I said before, is a consortium between 4 universities in Ireland, and there's a large number of people involved in these, so I'm here just representing that. And the 2 examples I showed you today were carried out by Africa Sullivan, Anya O'Connor and Brian Meehan, and the Department of Agriculture is funding both of these studies. So thank you for your attention.